Thank you, sir. Right. So today I'm going to talk about hybrid vigor and heterosis, where exactly this heterosis or hybrid vigor is going to come into place, especially in terms of genetics. So we are going to see that it is basically out crossing or hybridization techniques that are there, so that your hybrid vigor and heterosis will increase the first filial generation of progenies, performance. So it will increase the function of the biological quality of the offspring, which is a hybrid. And this genetic traits are much better as compared to that of the parents. So this is exactly something similar to that of the Mendelian or the non-Mendelian inheritances. So hybrid vigor is the improved activity survival of the hybrid offspring. So this phenomenon is basically something reverse of the inbreeding depression, wherein the vigor of the progeny is completely coming down. Instead, when it comes to the heterosis, you see that the performance and the functions and the traits of the progeny are much better compared to the that of the parents, and they often exceed the performance of the parents as well. So then this can be demonstrated in many kinds of inbreeds, in fact, wherein, the parents are either common or no common factor is present. So increase in the heterosis is usually seen or corresponds to the reduction in the common parentage. So because there are no common parentage present, the increased vigor is already seen. And then when inbreeding is imposed in the population as compared to the heterosis, you see that the genes that are there, especially those which are deleterious in nature, are going to come out. They are going to be manifested, and the weakened individual lines are also going to be coming into place. So when, if at all you take two inbred pure lines and you cross them, only the dominant favorable alleles that are there, they're going to suppress the recessive genes altogether, because of which you're going to get a superior line. So heterosis, you can actually see, is defined as the extra vigor superiority shown in the first filial progeny. And this extra vigor is not seen in either of its parents or the mid-parental value. And this is given by the formula F1 minus the mid-parental value by the mid-parental value into 100, or F1 minus better parental value by the better parental value into 100. So it's a percentage that is there. It was first observed in 1990 by a scientist named Shull in silkworms. And the progenies obtained by crossing two inbred lines showed better performance and extra vigor. So these factors which are going to determine your heterosis are your genetic relationship, compatibility, and the origin of the silkworm breeds. The theoretical explanation that you're going to see is basically, just a second, right, is basically that, there are four theories that are there. What is the genes that are going to control the characters? And then the theory of dominance. So if you see, basically, whenever there is a dominant gene, obviously that dominant gene is going to be manifested as a trait, and it is going to be in the phenotypic. So there is a phenotypic expression of it. And theory of over dominance and epistasis. Well, that is whenever there is a gene interaction as such, allelic interaction is seen. So if genes are going to be controlling your characters, they will obviously be controlling the characters as such, because they are the functional units of chromosomes, and all your traits are going to be determined by genes, whether they're single or multiple alleles, multiple genes involved in controlling that particular character. So output of the single gene controlled character is going to be less. And if at all multiple genes, for example, your multiple genes, multiple alleles, you can see in your ABO blood group system, in fact, you get an optimum combination. Theory of dominance, again, like I told you, if you have a dominant allele, it is going to be expressed in your phenotypic as the phenotypic character. Theory of overdominance, heterozygous dominant allele will have a stronger effect. That is to say, crossing of that homozygous recessive parent with the homozygous dominant. Simple. AA crossed, capital A crossed with small a. So you're going to get various combination. You're going to get A capital A, small a. So you're going to get different kinds of heterozygous. Small a is homozygous recessive again, like I told you. So heterozygous combination AA or small a capital A, you're going to get. So this overdominance can be seen. Epistasis is the interaction between the genes. It is going to take place when one gene is modified by several other genes. So you have something called as a modifier genes coming into effect. And then the phenotypic expression will be that of a different expression altogether in the case of epistasis. So usually you see that in flowers wherein you have red and white flowers. If they are crossed together, the genes are crossed together, you're going to get a pink. So that is one thing. So you have different kinds of examples. Even in silkworm, you're going to get epistasis, which will actually give rise to a different phenotype as such. So even though the different theories are all different, they're not exactly mutually exclusive. Only that phenomenon is not going to take place. So the phenomenon of epistasis, the phenomenon of heterosis is exploited in silkworm and in maize breeding. And usually even in animal breeding, pure line selection is used. So you see that epistasis and then heterosis, theory of overdominance, and then you have your genes that are going to control the characters, all of it, that is your, they come under the heterosis, and they're going to be economically useful in breeding better and superior silkworm breeds. So since the finding of the heterosis, many scientists, especially the Japanese scientists, did a lot of trials, and they crossed pure lines as well, and they tried to harvest the silkworms and better breeds wherein the silkworm the robustness was there, superiority in the production was there, cocoon quality, silk production, all of it increased many times by using the terminal crossbreeding strategy. And heterosis in Antheria asama, which is another like silkworm breed, Westwood in respect of this oviposition was observed. The mid-parental value was seen in respect of 4 crosses N into W1, N into W2, Y into W1, Y into W2. And then there is no inbreeding depression in the oviposition rate. That was seen till the 3rd filial generation. And cocoon weight and silk content in all the 6 crosses exhibited that heterosis over both mid-parental and better parental values was better. In the F3 generation, what happened was you could see a little bit of inbreeding depression for both the characters and the values that you are going to see, the quantitative values, and they were at par with that of the mid-parental value. So the depression that was seen and the quality and the quantitative traits were at par or equal to that of the mid-parental value, equal to that of the parents. So commercial exploitation of the hybrid vigor should be seen only up to the F2 generation if you always want to maintain the superior quality. That is what is being told about the heterosis. Now when it comes to hybrid vigor, it refers to your positive heterosis. So what happens is there is an improvement as compared to the parents, and the F1 progeny is much better compared to the parents or the mid-parental value of the genes or the alleles. So hybrid vigor is the manifestation of this interaction between the genes or the alleles of 2 pure lines. They can be inbred. So that's what it can, that is all right. But however, the phenotypic expression is always going to be dependent upon the genotype and the environment that is given. So dominant will give rise to better lines. Even if the organism possesses a good genetic makeup, phenotype is always going to be an interaction with the environment. So for optimum expression of the genetic or the genotype, you need a very good atmosphere for the silkworms to be present in. And this phenotypic expression is again dependent upon the genetic plasticity or the buffering of the organism. So whatever extra vigor is being shown is actually the output of the two inbred lines of the silkworm. That is because of the resultant organisms buffering that capacity, that is the adaptability to the surrounding environment. So during different stages of the silkworm, so many characters with respect to qualitative and quantitative aspects of the silk eel were present. However, the characters which reveal intense manifestation of the heterosis are like this. So whenever there is a duration in feeding, if it becomes shorter than that of the parents or the mid-parental values, it changes. Then your MPV changes. Mortality rate is lower than that of the other parents. And then you get to see that double cocoon rate is higher than that of the parents. And you have the MPV is basically your mid-parental value. So cocoon weight, double cocoon rate, cocoon shell layers, they're all heavier or more and better as compared to your mid-parental values. This includes the length of the silk fiber and the cocoon fiber weight over the MPV. So earlier results of two Japanese scientists showed that the following values of the hybrid vigor for different kinds of characters as compared to that of the mid-parental value as 100. So when this was taken, you see the silkworm larvae and the cocoons are being given, and this is the hybrid combination of APMW1 crossed with APS8 and APMG1 crossed with APS8. So one of the parents is the same. So heterosis is being given wherein the mid-parental value has been taken as or in index value as 100. So duration of feeding, larval mortality, double cocoon rate, all of this is different. So you get to see that duration of the feeding and the larval mortality is less than the mid-parental value, whereas your double cocoon rate, silk filament rate, size, cocoon shell weight and egg numbers are more. So this is very conducive and very economically very useful for the production of the silk. And in terms of different crossing systems, you see that hybrid vigor is the best in the first filial generation. So F1 is greater than F2, is greater than F3, is greater than F4 and so on. So we already have seen or have learned that up to F3 or F2, if we do the hybrid vigor and heterosis, it is going to be economically useful for us. Whereas if we continue for more than that, then you're going to have depression and it is going to be more or less same or lesser than that of the mid-parental values. So silkworm varieties can be crossed with single cross, that is A cross B, or you can also have a three-way cross wherein A cross B, the result of it is crossed with CJ, and a double cross pattern wherein A cross B is crossed with C cross D. So you're going to get lines which are crossed in this manner. So you're going to get a double cross pattern. Generally, whenever there is only a single cross hybrid, then the highest rate of hybrid vigor is going to be compared to that of the three-way and double crossed hybrid. And you're going to see that it is going to be high. Studies showed that variation in the quantitative characters, especially in the parental strains and the hybrids of the different, when you cross different systems, they show that variability for the quantitative traits is usually smaller in single cross hybrids as compared to the parental strains or the three-way cross and the double cross. So there's a paper that's been published by Watanabe in the year 1961 and 83 and 1973. So you can just check this particular paper as well. So when you summarize hybrid vigor or heterosis, this is basically wherein you're going to enhance your or by performing outbreeding, and you're going to enhance the functions and the phenotypical traits or the biological quality of the hybrid offspring as compared to the parents. Heterosis is defined as the extra vigor or superiority shown by F1 over either of its parents or the mid-parental value. And then when it comes to the phenomenon of heterosis, if it is fully exploited, it is fully exploited in case of silkworm and in terms of maize. Then in the case all the other animal breeding also, usually what happens is you select a pure line so that you can get a hybrid of your choice. Then when it comes to hybrid vigor, refers to the positive heterosis, extra vigor or improvement in performance shown by the F1 progeny over either of its parents or the mid-parental value. And this is good, or this is exploited up to your F1 or F2 generation only. If you do performing heterosis beyond F3 generation, you're not going to get much conducive results. So this is all about your chapter 3. I will have to start with chapter 4. I will try to finish it in this particular lesson itself. So we are going to check or see about different kinds of parental races. In this, we are going to see what or how important are your different kinds of races that are present. And then at the same time, how is it going to be the parental races based on the origin of the place where it's present, based on the voltinism, that is whether it is a univoltine, bivoltine or multivoltine, and based on the moultinism, how many times it is moulting. So we are going to just have a look at it basic. So you're going to see the history of silk. You see it's been more than 5 to 6,000 years. I should say it is like around 2500 BC, wherein sericulture was actually introduced, and in China. And Bombyx mandarina is a kind of wild silkworm which was reared in North China. So, and then after a lot of purification, today we get a modern silkworm which is there. Now, different kinds of climates are going to produce different kinds of races of silkworm. This will lead to be, for you to get different kinds of silk. In India itself, you have Muga silk from Assam, Tussar silk. You have your Mysore or crepe silk. You have different kinds of silk that are present only in India. Kashmir silk is also one of the um, types of silk that is available in India. But when you see, you have European silk, you have so different kinds of silk is there. This is all given by different types of silkworms. So on the basis of the, regions, silkworms are of four types: Japanese, Chinese, European, tropical races. So different climates produce and areas give rise to different specific silkworm races. European races are seen in Europe and Central Asia. Tropical or Indian are the native of India and Southeast Asia. Univoltine, bivoltine, multivoltine, like I told you, based on the life cycle, are usually found in Japan, China, Europe, and India. But multivoltine are more popular in warmer regions, usually seen in India as well. So parental races are usually present in that particular region in the world. So, and then this will, when combined with other races, evolve new hybrid varieties. And some of them are maintained as just stock or seed forms so that future generations can be derived from them. In Japan alone, you have around 2,000 genetically modified identified races maintained. This was seen in 1981, in fact. In India, you have around 200 races being maintained at different breeding centers. We have a central silk board which takes care and which is in charge of all the different kinds of breeding stations in India. And this is especially for evolving the commercial races or the multivoltine races. Now, when it comes to the parental races, like I told you, different silk races are present. Classification is based on the region that are present, the times of bolting, then the times of molting, the cocoon shape, larval shape, cocoon color, number of hatchings in a year, rearing period, body markings, cocoon color, egg color, etc. Now you get to see China, univoltine China. It has given rise to China bivoltine. This has been giving rise to the tropical polyvoltine. Again, univoltine type or silk breed has given rise to your European univoltine. And this again, China univoltine has evolved or has given rise to Korean univoltine, which has given rise to your Japanese bivoltine and Japanese univoltine. So this is all about your basic parental races. Now, based on the place of origin, there are four types of races, as you can see, which are Japanese, Chinese, European, and tropical. So again, based on the different types of races, Different kinds of larval, pupal, and adult forms are seen, and all the biological characters that are present, that is life cycle, diapause, the times, number of times they are molting, environmental factors that are going to influence them so that they give the filament length, denier, reliability, mortality, shell ratio, all of this are going to change as per the place of origin and the race of the silkworm that is present. So when it comes to the Japanese race, you have larvae which are strong. They are resistant to different kinds of extreme environment. But the larval period that is there is long. You have both univoltine, bivoltine silkworms in the Japanese race. Eggs which are of different colors. They may hibernate, they may not hibernate. Many white rotten eggs are also going to be produced, because of which the eggs are going to die after pigment stage itself. Now, whenever the larvae are healthy and strong, they are going to grow slowly. Markings on the skin are going to be normal, but some of them sometimes show quail markings. Body is black in color. Larvae are yellowish during the molting time and red during the ripe stage. So the larvae are going to be slow in eating. So like I said, larval period is comparatively long. So worms are not going to identify the leaf quality. So you have a type of disease that is going to come and attack them, which is your neurofibromatosis disease. Cocoons are going to be dumbbell shaped, that is the 8 shaped, or peanut shaped, or spindle shaped with white or straw color. Some of the races are going to be green or yellow in color. These races produce more of them. Whenever there is a double cocoon present and slightly short and thick cocoon filament present, then you're going to have this kind of phenomenon is quite common. And the cocoons are going to be inferior in quality with fiber length which is smaller, and the reeling is going to be generally poor. So you get, when you're doing the reeling, you should see that the reel or the thread does not get cut off. So the reeling quality is very important over here. So these are the characteristics in case of the Japanese race. In terms of Chinese race, the Chinese silkworms love to eat the mulberry leaves very actively, and the progress of the larval growth is very fast and it is very uniform in nature. Larval marking on the of the many races from the Chinese silkworm, or the Chinese silkworms are in fact plain, and they are not sensitive to high temperature and muscadine, but sensible to the high humidity. And these eggs are slightly yellowish in color. And like I told you, if it is sex limited, wherein the females are yellowish in color, the male white, or vice versa, it is very easy to know or distinguish between the cocoons. So larvae are white with round body form, white during molting time, blue in the spinning stage. They grow rapidly by feeding actively on the mulberry leaves. They are trimolters, meaning they molt 3 times. They cannot identify the surrounding temperature, humidity, and air. The cocoon form is also oval or spindle shaped and are white, golden yellow, green, flesh colored, red or even pink in color. And the cocoon filament is going to be long. You have a good reliability coming, long, thin, and you have univoltine, bivoltine, and polyvoltine races in the case of the Chinese race. So you have few trimolters also, which are like, and most of the races are tetramolters. So these cocoons are either uni, bi, or multivoltine types. When it comes to the European race, mostly univoltine eggs are big, heavy, dark brown in color compared to the other races. Hatching is again irregular. Larvae form are like, big, long. They have pale normal markings. Larvae are yellowish during the moult, red in the ripe stage. Larval marking is lightly normal, and silkworm larvae eat the mulberry leaves actively. Duration of the larval stages, especially the 5th instar stage, is long, and you have a long larval body as such. But then these larvae grow fat quite easily because of the complete ingestion of your mulberry leaves. Slightly difficult to rear. They are sensitive to bad environment, pebrine, muscadine, and the different types of C-type virus disease. So they are again weak against high temperature and humidity. They spin large and have a long oval cocoon. So you can see this is the type European race cocoons that are present. They are pinkish in color, white or flesh colored or yellow in color as well. Cocoon shell weight is quite good. Cocoon filament is long and you have a lot of sericin and good reliability is also seen. So double cocoons are less. You see the Japanese race is like peanut shaped or it has an eight shaped. Chinese race basic oval shaped and this is the Indian race. So usually the European races are univoltine in nature. And the tropical races are multipotent in the case of tropical the race and they have non-hibernation or non-diapause eggs. So so that what happens is during hibernation the status quo is maintained and an egg white or of the type 3. So you have W3 gene that is present. The eggs are small, they weigh light, and they have a lustrous shell as such. And the silkworm larvae are small, and again they are going to be long, and they are quite healthy, that is robust. They're tolerant to high temperature and humidity. And the larval duration is comparatively short, except for your pure Mysore race. These races again produce a small cocoon, you see, with spindle shape and they can be green, yellow, or white in color, and they can be flossy as well. So the cocoons that are loose and flossy with light cocoon shell weight and thin cocoon filament. Double cocoons can be present. Sericin percentage, that is your protein percentage of these races is quite high, that is around 30%. So what happens is raw silk percentage becomes low. However, the cocoon filament is fine, thin and clean with little lousiness and the disease that is seen is muscardine. We also have a Korean race. In the Korean race, they're all univoltine and trimolters and larval duration is comparatively short. And again, it is a small cocoon that you're going to see with a low cocoon shell ratio and the cocoon color is almost yellow or yellow-green. Few are white and you have a short and thin cocoon filament. Now you have the regional races that are present and you have a comparative chart that is present showing your general characters of the regional origin races, Japanese, Chinese, European, Korean and tropical, the different kinds of larval markings, the larval duration, voltinism, is it univoltine or bivoltine or tetravoltine, and then the cocoon shape color, you see Japanese is peanut or spindle, Chinese is ellipse or oval white flesh, European is long ellipse, Korean is spindle, tropical is around small white green. So you have different colors. The type of silk that is going to be there, thick, thin and long, long in good reliability, thin, flossy with thin cocoon shell. So now based on the voltinism, like I told you, we have univoltine, bimoltine and multi or polyvoltine. So that is, you're going to see the ability of the silkworm to produce one to several generations in a year. This is known as your voltinism. So when it comes to univoltine, silkworms that are producing only one generation in a year are called univoltine, suitable for cold regions. They lay diapause eggs, that is hibernated eggs, due to the absence of a hormone called as a juvenile hormone. Now, larval duration is long, larval body size is long, cocoons are large, cocoon filament is good quality, but you cannot grow this univoltine in the hot season because the larvae are not resistant to the bad conditions such as high temperature and humidity. They are unsuitable to grow in summer and autumn. Diapause eggs are reared by artificial breaking. Cocoon filament is good quality. Shape of the cocoon is round or oval, which is white, pale yellow in color. When it comes to bivoltine, you have two generations. Again, the first generation does not lay non-diapausing eggs. There is no break. Second generation develops from the non-diapausing egg which was laid during the first generation and that lays the diapause eggs due to the absence of the juvenile hormone. So the second generation is going to be dormant till the next spring and it comes out of the first generation. These races are suitable for the temperate zones where the temperatures are very conducive. The larval duration is short compared to univoltine and the larvae are robust. They tolerate different types of environment. In fact, cocoon quality is inferior as compared to that of the univoltine races. But then better, or you get to see that bivoltine races also have good commercial value. So that is more important over here. They can be reared during the summer and autumn as well. Please give me a minute. Yes. So the leaf cocoon ratio is less, and cocoon weight, shell, silk percentage, filament length they are all going to be lesser than that of the univoltines. Cocoons are usually dumbbell shaped or oval in shape, white or pale yellow in color. When it comes to the multi-voltine, so you have 5 to 6 generations that are being produced in a year suitable for the tropical regions. So hot, wet, humid conditions. Duration is again short. Larvae are quite robust, tolerant to high temperature changes. Leaf cocoon ratio is high. Cocoons are small in size. Percentage yield of the raw silk is poor. However, the cocoon filament is thin in size, clean with a little lousiness. So you can see good cocoon size. They produce the inferior quality silk as compared to that of the uni and the bivoltine races. That is why China silk is really good. But the shell ratio is less again, and the filament ratio for the filament length is short, and the filament is fine and clean with little lousiness but with more luster. But owing to our silk industry, I think this is much more suitable for us. And, it is really good that, we follow or we have indigenous silk and your silk board, which is doing a very great job. So the larva forms are robust, can tolerate fluctuating environmental conditions and hence best suited for the tropical climates. They lay only non-diapause eggs due to the presence of juvenile hormone that is present. Example is your pure Mysore, then C. Nietzsche is one, Hosa Mysore is one kind of breed. When it comes to Maltinism is a very important economic character along that of the voltinism. So 5 types of molting is seen in the silkworm. Dimolting, trimolting, tetra, penta, and hexamolters. Most common type in the domestic silkworm varieties are the trimolters. So they mold 3 times. Whereas almost all the commercial strains that are there, which are giving you your commercial silk are, or they're going to mold 4 times. So tetramolters. Tetramolting character has been considered as the standard in the silkworm genetics. So the expression of the molting character is also going to be affected by your sex-linked maturity genes, that is L and M, LM. So dominant relations are in such a manner that M3 is greater than plus M greater than M5. So trimolta greater than plus M greater than M5. Manifestation of this molting characters is also controlled by balance of this hormone, that is the juvenile hormone that is present in corpus allata and ecdysone of the prothoracic gland in the silkworm. So you have different kinds of segments in the silkworm, right? So you have the prothoracic gland, and ecdysone is the molting hormone that is present. Ecdysis is the phenomenon that is equivalent to or that is known as your molting in common terms, but molting's scientific name is ecdysis. So that is where ecdysone, the hormone, comes into place, which is present in your prothoracic gland. Now, silkworm larvae, they cast off the old skin, they develop a new skin, and this process is known as moltinism. And this molting period lasts around 15 to 30 hours. And it is a hereditary trait. Molting is a physiological process, and this is under the control of your hormone, which is ecdysone. So, based on the number of molts, the Bombyx mori is a tri or a tetra or a pentamolter. Now, in trimolters, length of the larval stage is short. Larval body cocoon is small. Cocoon filament is fine. Tetramolters, again larval duration is medium. Produces a thin fiber. Most of the commercially reared worms are tetramolters with 5 instars. In the larva pupal form itself, you have instars also present. So most of the commercially reared worms are tetramoltas. They molt 4 times with 5 instars. Five, and these worms are most widely reared. In pentamoltas, the larval length is long, body cocoon, and the filament length is going to be larger in size. So if we summarize this particular topic, you see that parental races are going to be originate from different geographical parts or are of different geographical parts. They are Japanese, Chinese, European, and the Indian races. And these races are again classified based on the native origin that is present, number of times they molt, which is moltenism, number of generations that they produce, that is volatinism, the cocoon color, and the larval marking. Again, races can be differentiated from one another on the basis of morphological, biological, and commercial characters. So, Japanese race gives rise to non-hibernating eggs. Dava grow slowly. They're usually black in color. They eat slowly. They're inferior cocoons. Chinese races are again robust. They're healthy, tolerant to high temperature. They grow rapidly. They molt 3 times and they give good reelable silk and silk filament, which is long. And when you see the European races, the eggs are big, heavy, and irregular in hatching. The larvae are long, and you have a long life, and the cocoons grow fast, but they are resistant. They are not tolerant or weak against the high temperature. Cocoons are long again with more shell weight, and they have a long fiber and good reelability. When it comes to our tropical Indian races, the larvae are small and they are tolerant against temperature and humidity. The cocoons are going to be shiny or flossy, but they have less shell weight, but the filament is going to be fine. And again, based on the number of generations produced in a year, the silkworms are of 3 types. They are univoltine, bivoltine, and multivoltine. We have the characterization again based on moltenism, which is the number of times they molt. So they are trimolters, tetra, and pentamolters, wherein they molt 4 times, 3 times, 4 times, and 5 times. This molting is again based on or dependent upon the hormone, juvenile hormone, and egg disone, which is produced in your prothoracic gland of the silkworm. So this is all about today's class. I will end it over here. Tomorrow we will have a look at the different kinds of breeding stations and we will have a look at the rest of the topics.